So why model the brain? And so, of course, this I hope since you all have signed up for a school on how to model the brain, I guess you have some ideas about why you want to do it. And I like to start to like showing the picture of this guy. He's on a Norwegian stamp, Niels Abel. So he's a very well-known mathematician and probably Norway's most famous mathematician. But he died in like 27 years old. You can see that in the lower right corner of the stamp for tuberculosis. And he was so upset about the medical doctors at the time. Why did they have to die so young? Why couldn't they fix this thing? Of course, now today you don't die of tuberculosis in at least in our part of the world anymore. But if you go to the doctor and come back with a serious brain disorder, it could be like a psychiatry or like dementia or something, it's very little that medical science can do. And this is also reflected in this paper that was written in 2012 by this Stephen Hyman, who used to be a director of the National Institute of Mental Health. So what it essentially says there is that why are we doing so poorly? Why, and this has had to do with psychiatric disorders, why, despite all this research and all the money that has gone into this kind of research over the last decades, why are we doing so poorly? And even like major pharmaceutical companies have stopped doing research on some of these brain diseases simply because they haven't made any progress. So why are we doing so poorly? Of course, at least when you're doing poorly, despite a lot of effort, like we've done in research, you can ask yourself, well, are we doing something wrong? Should we do our, in this case, research differently? And if you ask neuroscientists, what is the problem? Well, they often point to diagrams like this, showing the different scales in the brain. That it's like you have all the way from molecules, proteins, that's the molecules of life, all the way up to like the cells, the neurons and the circuits, all the way up to the whole brain and the whole body. And you can also look at the temporal scales that spans many, many decades, all the way from the time scale of molecular interactions, all the way up to like milliseconds of the typical time scale of neural signaling, up to the lifespan of the human. So they have all these scales, and they are intimately connected. It's not like they live completely different lives. And that's a feature of biological system. For example, a thing like eye color, which happens then at the centimeter scale, that's the size of eyes, can be regulated by a single molecule or a single like element of the DNA molecule. You can switch one gene or one amino acid, and you end up with a different eye color. So that is a little bit discouraging. Why? There's all these scales. Typically, neuroscientists work at one particular scale, and it's been difficult to bridge these scales. So, but are there like fields outside neuroscience which maybe have something to teach us? Well, if you look at weather prediction used to be these very sort of like what I would say approximate, like rough science, where we had some like weather science and stuff. But then, the problem itself is quite complicated, and it's complicated in a similar way as the brain is, in the sense that there are many scales involved. You have all the way from like the 10-meter scale, with like turbulence and stuff, going all the way up to the size of the globe. And a mathematician, a Norwegian meteorologist, said about 100 years ago, a little bit more than 100 years ago, that the weather prediction is a mathematical problem. So what it meant was that we know at that time they knew all the basic laws of physics, and like with temperature and pressure and wind and so on. So if we know the weather today, meaning the temperature, pressure, and so on, at the grid, covering the well, the Earth, you could in principle compute the weather in the future. And this was, of course, at the time, not really possible in practice, because this is not the kind of calculations you can do by hand with pen and paper. You need strong computers, and that you didn't have at 100 years ago. But now we actually, well, now, I mean, the first weather forecast from mathematical models came with the advent of computers in the 50s. And now all weather forecasts are computed from such models. So it's like a steadily improved quality also. These weather forecasters predicts one day better into the future per decade. So the accuracy of predicting 6 days into the future today is about the same as predicting 5 days into the future 10 years ago. And here's actually the weather forecast I took from my smartphone from like my area around my house a couple of days ago. So we all use this, and they are getting better and better. And that's all due to mathematics. There are also other examples where we have used maths to bridge scales, and smartphones is one of them. I mean, if you look at the smartphone, you can sort of, it also has many different scales, and these scales have to work together. It has to be connected. And if you look at the scales of the brain, you can start with the atom. Everything are made of atoms, whether you're a smartphone or a brain. And then you have molecules, molecular networks, all the way up to nerve cells, neurons, and then neural circuits up to the brain. And you have the same kind of scales, or like roughly the same time, well, similar scales for the smartphone, atom, crystal, these tailored materials like semiconductor heterostructures and metals and stuff, the transistors, which in some sense is the fundamental computational unit in the smartphone and chip, and up to the smartphone. So how did the smartphone people do it? How, I mean, not a single person cannot build a smartphone. But what they found out, well, what they could use was they had this very precise language for engineers and scientists at these different levels to communicate. And the key language that was important there, you need a precise language to communicate, and that's mathematics. So, and it's also actually, when you started to program these things, you also needed a very precise language or different kind of computer languages to make sort of like, to make the connection between, like, if you want to say, if I sort of push a button to see my local weather, I'm actually in some sense programming in a very like a sort of like overarching like simple computer language, some sense, but that has to sort of connect then to computer programs at the levels of the transistors. So you need precise languages to be able to bridge these scales and communicate. So what's the situation in the brain? Well, actually, we know how to model essentially the physics or the chemistry, whatever, nature at these different scales. So we have like the quantum mechanics at the atomic scale, and you have rate equations and so on. And at the neuron scale, you have some ways to compute things based on the cable equation and something called multi-compartmental modeling, which Bruce will tell you about afterwards. So, and then also at the neural circuit, we have like ways to model activity at that level, but we don't really know how to connect them as of yet. But the good thing is we know that the principles of brain modeling are known. I mean, we know how to model like single neurons, and also how to put them into networks, and we also know how to connect sort of like what you do make in a model, say a network model of a piece of the brain, to what you would measure at say with the measuring the electrical potential at the scalp, for example, with the EEG recording and so on. So we have all the elements, at least, or we know the elements of this program well enough to be able to actually to use it to explore the connections and to build these bridges between the different levels. And fortunately, we have some really excellent work and some excellent models to build on. And I think the breakthrough was really this work by Hodgkin and Huxley in the early 50s, where they found a way to model how action potentials, these pulses that neurons send to each other, and model how they sort of are generated based on the basic biophysics of these nerve cells. It has to do with sodium current, particular sodium currents, potassium currents, and so on, which goes through dedicated ion channels going through the membrane. And these ion channels are little tunnels, and they are made by proteins. These dreadlocks here are proteins going up and down through the cell membrane, so that making a little tunnel that allows these green balls, which are potassium ions, let them through, while actually the purple balls are not allowed through. That's the sodium ions. So that's just illustrating sort of a key feature of these nerve cells, that they have ion channels that are selective, meaning that they let through only one type of ion. So based on that, we can make simulations like this. This is just a simulation made by Espen Hagen in our group, and just illustrating, I guess there are 4 neurons, because each of these neurons are this beautiful branching structure. The color here represents the membrane potential. That's the potential difference between the inside and outside of a cell, which is about in the rest, it's about -60, -70 millivolts. But you see in these, and that membrane potentials vary across these neurons. And what you can see here is that sometimes they flash. And that's when they fire these action potentials, because then you get this depolarization that maybe lasts for milliseconds or so, and they send the signal to other neurons. So this is just, of course, just to illustrate the principle that we have. In this case, we can simulate neurons. We can put in things in our model and explore how the model that we suggest, how it behaves, and then compare with experiments. When it comes to networks, we are doing we are not we have a solid starting point. We know how to model neurons. At the moment, we haven't made much, or at least we don't understand really how neural networks work, meaning we know how to build models, network models. We know all the ingredients, we know the neurons, how to model them, we know how to model the connections, but it's very difficult for different reasons, it's difficult to actually to make network models that corresponds to particular biological systems, like say, for example, a piece of a mouse visual cortex. And that's one of the things that our group here in Norway works on, in collaboration with the Allen Brain Institute in Seattle, who has been focusing on the mouse visual cortex for almost a decade now. It takes a world to understand the brain. That's the thing. If you want to model the network, it's one thing to have an idea about, I think this network is the basic principle is this and that. But you need, in order to compare with experiments, you need to be able to have model predictions that is sort of to compare with experiments. And in order to, these are sort of models that are so complicated, or I mean, if you want to model, say, I mean, the human brain has 100 billion neurons, but even if you want to model a mouse brain, it has 100 million neurons. But even modeling like 100,000 neurons of these sort of like biophysically detailed neural models requires really strong computers. And it also requires an infrastructure. I mean, it requires really a collaboration between scientists, both to build the models and to run the models. So we are involved in the Human Brain Project, which is this European initiative to work on that, to build the sort of like to build the infrastructure necessary to essentially so that you can sit that mathematical neuroscience, that you can sit anywhere in Norway or anywhere in the world and actually be able to simulate pieces of the brain use running programs on computers at like certain supercomputer centers in all across Europe. And there are also other projects around the world trying to sort of to make these, well, to make it possible to simulate networks. So we wrote a paper on sort of why we need to do large-scale brain simulations, and it's not because we think that like brain simulations is going to be sufficient alone, sort of to, it's not like you just put a network model together and voila, out comes a smart brain. That's not how it works. You need these large-scale brain simulations in order to test out your hypothesis. So it's more like a mathematical observatory for how networks may work, and then you can have model prediction that compares with to be compared with experiments. So I think I'll just you also need that to model like you need sort of to make a connection between your model and the things you can measure, because there are many measures of neural activity. The traditional one is to put down a sharp electrode and to measure the action potentials, these pulses, and they are typically done by having an electrode that, at least in living brains, that sort of measures this action potentials from immediately outside the cells, something called spikes. But each spike then corresponds to this neighboring neuron firing an action potential. Or you can model the membrane potential. So that is sort of if you're able to impale the cell so that you can model the potential difference between the inside and outside. But now we also have many other types of electrodes that is not only can sort of measure these individual action potentials, but also other aspects of the activity. For example, if you put down an electrode like this with all these circles here correspond to individual electrode contacts, then you can sort of, each of these contacts will be able to record both of these action potential, but actually most of the power is in the low frequency part of the signal, what's called the local field potential. And that reflects other aspects of neural activity, mainly how sort of the inputs from other neurons are processed by neurons, while the spikes more represents the output. And then there's also a bunch of optical techniques where you shine in light and you put in different kinds of ink, and then you can, depending on what kind of ink you have and so on, and what you measure, you can sort of infer neural activity from these from what comes out. And then of course we also, in addition to this, measures inside the cortex, which is illustrated here. This is sort of the cortex with a bush of neurons. You measure LFP, local field potential, and spikes with a single electrode like this. But you can also measure potentials, electrical potentials at the cortical surface, or even better, at least if you want to use it on humans, is to measure the EEG or the electrical potential at the scalp, or maybe the magnetic field outside, because these are non-invasive, so they can be used on humans without any troubles, while these are invasive and they are never done on humans unless there is a sort of like a clinical reason for it, that they have to do these recordings in order to prepare surgery or something like that. So if you have these models, then you need to, it's like a very simple sort of standard approach in physics and engineering. You make models, and you make models at different levels of detail, because these different levels of, simplified models have their advantages. They're more easy to deal with computationally. They're also more easy to understand. But you need, the more detailed models are often easier to compare with experiments. So then, if you want to make a model for a network, you have these models, put your hypothesis into these models, and then you take your models and compute, okay, if this was the world, what would you measure? And then you do predict all these things you can measure, and then you go back and forth to sort of iterate yourself to the sort of like the model which is maybe closest to experiments. And so then, in order to do this second step, you need to be able to link the activity in neurons to these different measurement modalities. And so each of these measurement modalities reflects different aspects of the same neural activity. But if you have a good biophysical model for a neuron, you can compute all these things. So you need to work out these mathematical connections between the neuron dynamics and the different experimental modalities, sort of the measurement physics. And here's an example from our group where we have developed this tool LFPy to do this. And with this, this just illustrates what the tool can do. You can put, say, like a 10,000 biophysically detailed neural models in and drive them with input from other neurons. And then from that, you can compute the spikes. You can compute what you would measure the potential, the low frequency part, or the extracellular potential you would measure with electrodes going through the bush. But you can also compute what you would measure at the top of the skull or with the EEG recordings, electrical potential, or magnetic field outside. So the point is that with this tool, you can take any kind of model you want, your favorite idea for how this piece of this cortical circuit is hooked up, and then compute all the things you need to compare with experiments. Let's see. And here's another example where we combine it with compute also the LFP and EEG and also spikes from a network formulated as simpler neurons, like called point neurons, where it's not like this, each neuron have these beautiful branches and so on. They're just a single point. And they just follow from the basic measurement physics that these do not generate extracellular potentials like the LFPs. So we need this trick where we sort of first compute the action potentials, the spiking with one kind of scheme using nest, and then replace each of these, in this case, 80,000 neurons with a multi-compartment neuron and replay the whole thing, and then you can compute the LFP. So here is sort of an example of this, where there's just a network running at a time 900 milliseconds. Then there's an activity, this is a piece of the visual cortex. So at time 900, it gets this input from the thalamus, which will correspond to visual input. And then what you see then, if you come, next time you come to 900, you will see that actually there's this activity, spiking activity there, and followed by a silent period. And at the same time, you can actually see the signature for the local field potentials. This is what you would measure then with the local field potential. And this is what you would measure with the EEG electrode at the top of the head. So, and this, of course, then if you have all these measurements and you can compute these things, you can sort of, if you have different network states, for example, or different models, each state here corresponds to different models in terms of the different choice of parameters. You get different, you can compute and get all these signatures. And then if you have experiments, you can sort of compare and say, well, I think actually my experiments is closer to network state 3 than the other two network states, for example. So now, finally, I'm just going to say a couple of slides about this project that we're working on. This, the state-of-the-art model of a mouse primary visual cortex, 250,000 neurons, and it's really been built on it's essentially based on almost 10 years of excruciatingly detailed and almost industry style approach to mapping out all the neurons and the connections in this circuit at the Allen Institute, and it's been put together then into a very detailed model, and with different neuron types and so on, using all this bunch of different techniques. And then they made this model for the primary visual cortex of mouse, which we then are now collaborating with them on trying to actually make this sort of, I would say, almost what we call a skeleton model, because the hardest thing, well, there are many hard things. It's hard thing to put up the network, but it's also very hard to find out all the parameters that you don't know, sort of how neurons are connected and so on. So this is something we're working on, trying to make contact and maybe make this, develop this model into what we call the first multi-purpose model of a piece of cortex. So that is a model that can predict many types of experiments at the same time. So then we use both compare the model predictions with experiments of spikes and also the local field potential. So finally, is I'm not quite sure if you're interested in this, but maybe some of you are. It's a free online workshop where we talk about how to make this such multi-purpose neural network models that I'm involved in organizing, and that starting is next week. I think it's next week. Yeah, next week. And it's at if you want info, you just go to eitm.org. That's European Institute of Theoretical Neuroscience.org, who is doing the virtual hosting of this, and this is a virtual network. So thanks. That's what I plan to say.